5' and 3' directionality or polarity. I'm going to not use the term polarity. I'm going to use directionality. So there's the triphosphate at the 5' carbon. At the 3' there's the OH group. At the 2', if it's DNTP, there's no OH. And that's the building blocks for DNTP. Sorry, for DNA. So we come in here, we'll form this bond here, a phosphodiester bond. We have a pyrophosphate as a leaving group, and that gets broken down into 2 phosphates. This releases a lot of free energy. So this is driven by entropy, a lot of energy release, okay? And we see that we just extended this by 1. And this will keep going as long as we have the template. Okay? Any questions on that? Okay, so this is how traditionally it used to be done with radioactive, and then we'll talk more about more recent technology. We have 4 samples like this, and in this sample, it's labeled dideoxy ATP. It has all 4 types of deoxys, DATP, DGTP, DCTP, DTTP. We must have those if we want to make a polymer. But we have a small amount of dideoxy, a ratio, and the ratio is about, and it's not exact, but usually we have a ratio of dideoxy to the deoxy form of about 1 to 300. And that's a ratio. Not 1 molecule is not 300 molecules, it's a ratio. So these are large numbers, but the ratio is this. Okay. And then we have set up 4 tubes like this. And what varies here is the dideoxy in each of these tubes, okay? Now, I'm going to get the analogy first, and hopefully this will help. And I'm not, some students always put evaluations saying I'm not politically correct, but we'll just do it anyway. Let's pretend this is the dining commons, and there's 1 million students in each room. You know how it is, it gets kind of crowded. But these are very homogeneous students. They all weigh 100 pounds, every one of them. Okay? And I'm going to call this the A room, the C room, the G room, and the T room. And in each of these rooms, they get a stack of 301 cards. 301 cards. 300 of them say eat. 1 card says stop eating. Okay? Randomly shuffle them. So in this room, 1 million people have randomly shuffled those cards. And the announcer says, everyone eat. And then they say, hey, those of you in the A room, pick a card. And a subset in this room get the card that says stop eating. And they will weigh 101 pounds. And that ratio is about 1 out of 301 of 1 million. But everyone gets to keep eating. So after 2 minutes, the announcer says, hey, those of you in room A, stop eating again. 102 pounds. And then room, another minute, C, they say stop eating. 103, 104, 105, 106, 107, 108. Does that make sense? Seriously, let's do this. 4 rooms, they eat all the way 100 pounds, they keep eating. These rooms keep eating for that full minute. This, why? Because I'm doing an analogy. Okay, they don't have a choice, all right? Anyway, since in this room I say pick a card, 1 out of every 301 ratio-wise of those students get a card that says stop eating. Is that clear? Do I need to go over why that is? No, excellent. So they weigh 101 pounds, but everyone else kept eating as well. So everyone else was 101 pounds, right? And so after another minute, I say, hey, those of you in room A again, stop. There's a subset here now that weighs 102, and in all the rooms they weigh 102. Is that clear? Then a minute later, I say, I'm not going to pick on room A anymore. I'm going to pick on room C now. 103, 104. Is that okay? And then I say fire, and they all run out. And the people who weigh 101 pounds, they go quickly. And they come out of the A room. And I see 101 pounds. And then I see the 102 pound people, they come out a little bit later. And then the C room, 103, 104, T, 105, 106, G, 107, 108. Do I need to go over that? Now, why do these come out fast? Because they don't weigh as much, okay? Now, if I was standing out in the street, not knowing anything about the time column or anything like that, I could recreate what happened by just looking at this, right? I would know that I first called this room, then this room, then this, then this, then this, right? I could figure out the exact order of what I called them in and which room, right? As long as I know the labels here. So it's going to be the same way for the sequencing. Similar principles, a large population of identical molecules that extend for a certain time, and a subset will stop because of the ratio. All right, now let's look at the science. All right, so now that we have this, so in this dideoxy A tube, as we extend on this template, where will the decision for dideoxy versus deoxy take place? What will be in the template? What will be opposite it? Will it be a C? No, what would be opposite for that either dideoxy A or the DA? A T in the template, correct? So in this tube, we will get a subset to stop whenever it's complementary base pairing to a T. So the dideoxy A looks like this. So in the, I don't think there's a board here. Oh, there is, excellent. There's the CH2. There's the triphosphate. This is the 5 prime, 4 prime, 3 prime, OH, 2 prime, 1 prime, the base is coming off of here. This gets the solid numbering. Since this was a deoxy, if it had a ribonucleotide triphosphate, it would be like that. A deoxyribo is like this, and a dideoxy is like this. So can I extend, once this has been incorporated into this Roaring strand, I can't extend anymore because there's no OH here. I can't form that phosphodiester bond anymore. So whenever a dideoxy comes in, I will stop the chain elongation. I can't synthesize anymore, and then I will be fixed at a specific size. Okay? So in that tube, there will be products of specific sizes, and those will correspond to in the template wherever there's a T. Okay? So the same is true to the dideoxy C tube, but what would be complement to it in the template if it's a dideoxy C? A G. And for the T tube, what would be in the template? A. And in the G, C. So we can figure out the sequence. So looking at this, let's think about this. This is going to be the smallest, because when we run the gel, the smaller pieces of DNA will migrate fastest. They migrate fastest because they're the smallest. So that means these are the smallest pieces, these are the largest. Just like here, the smallest ones went fastest when they exited from the room. In this case, we're sieving it through a gel, but again, the same thing is happening. The smaller ones faster, the larger ones run slower. They're higher up on the gel. Okay? And DNA is negatively charged, so we just put a positive pole there, and they help create that force. So we see something like this. And we see bands. These bands are due to the fact that we've made the dideoxys or the primers either way. Let's say the primers are radioactive. So we see bands there that correspond to this sort of information. So in this, let's read the sequence from the bottom of the gel up. This is in the T lane. That means there's a dideoxy T there, right? At the first position. What's in the second position? A T. What's in the third? G. What's in the fourth? T. Fifth? C. Sixth? Notice I'm numbering from the bottom up. So this is number 1, the smallest, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Does that make sense? So the moment I step at the board, I can no longer see this. So someone read off the sequence to me that I should have. There's a T. Is that correct? And then another T. This is in the molecule being made. Not the template, but molecule being made. And we know there's a dideoxy T there. We know there's a dideoxy T at that position. And then what do we have next? Third is a G. Is that correct? What's at the fourth? Fifth? Is that it? Okay. So that's the strand that was being made. And synthesis is always 5' to 3'. Synthesis is always 5' to 3', which means this is the 5' end of that, and this is the 3' end. Okay? All right? Question? Okay, so the question was, if this is for the primer sequence, why isn't it the opposite? I actually haven't talked about the primer. The primer would have allowed us to start this, so that's the primer there. This is the new sequence there, okay? But, so I think the question that you'd like to ask is, can we figure out what the template was? And the answer is absolutely, because the template is 3'. What's here? A, C, A, G, C, T, T, C, A, G. So the gel is based upon what is being made, which tells us also information for the template, okay? Now, this is something you need to go back and look at and think about, okay? It's like those math problems. Kind of makes sense, but go ahead and try it, okay? So anyway, we'll move on. So here's an example here. This is what you might find on the lab exam, and you see a pattern like this, and it just says, what was the sequence of the strand being made? Or I could ask you, what was the sequence of the template? But remember, you have to label the ends 5' and 3', okay? Now, if you're wondering why is this the 5' end, remember what I did? We didn't know the template. The template was there, but we didn't know it. So we had a primer over here, which had the 3' end, and then I attached this one coming in, 5' to 3', right? And that made a 3' end. In the dideoxy, it didn't, but in the one that allowed this dideoxy, it had the 3', on and on. So this is going this way, 5' to 3' growing. That means this has to be 3' to 5'. Okay? So go back and think about it a little bit. I think that's the part that actually tends to trip up a lot of people. All right? Now, the way this is done is not typically using radioactive molecules anymore because it's not the best for the environment, things like that. Also, it can't be very well automated. So instead, we now use molecules here that are fluorescent. So the dideoxys themselves are fluorescent. So what happened then is whenever we see in this population here, so I'm going to just redraw this. Everyone clear on the dideoxy? Why that stops? Because there's no OH here to attach to. I just can't attach. So what this population would look like in all those tubes, in the dideoxy tube, there'd be the primer plus the dideoxy T, there'd be the primer plus a T and the dideoxy T. So that's why we would have seen in that gel, one at the 1 position, one at the 2 position, another one at the 4 position. But when we run it out in the gel, we see band at 1, band at 2, band at 4. When we put the information together, we said a T there, a T there, a T there. And in the G lane, we would have had something that looked like this with the primer, and then we would have had a DT, because we had a low extension, a DT, and then a dideoxy G, which is why we would have seen one at the 3rd position and also another one at the 6th, okay? So please try to draw that out. Make a template, separate it, draw what the band should look like, and try to figure out what's going on at each point, okay? It will help you, okay? And I lost my laser pointer again. But I have a backup, I think. Okay, so now let's think about this if this is fluorescent. So now when we have the dideoxy C, and this is 5 long, if we have a laser here, and that sample's in the gel migrating, but it's only one sample, so we don't have to do 4 different tubes. Here we had to have 4 different tubes. In this case, since the dideoxys are fluorescently labeled, we can get by with 1 tube. And each of these is fluorescently labeled. So when we have the dideoxy C stopping this, in that tube, we have a molecule that's 5 long, and we have another place that's this long, again with dideoxy C, and we have a dideoxy G that's 6 long, another one here that's dideoxy G, dideoxy G. So the one that's smallest migrates first past the laser, and because this is fluorescently labeled, as it migrates past, moving downward, the laser sees a pink color, and it detects that. And a little later, the one that's 6 long has migrated, it sees a little green. And then a little bit later, the one that's 6 long, or 7 long migrates, it sees the purple. So we get a color trace throughout this. This is much more efficient because it can be automated, and we don't need to have 4 separate tubes, and we're not working with radioactivity. So this is the method that's usually done. Question in the back. So this had to be a DT because we had to extend it till it terminated here. If this had been a dideoxy T, it would not have worked. Remember, in order for us to keep extending past this, we always have to put deoxys in, right? But when we stop, it's a dideoxy. So everything to this side of it has to be the DTs. Does that make sense? So on the left side here, this was the primer, and I had mentioned that one of them stopped at the 1st position because that subset, 1 out of 301, got the dideoxy T. But the other 300 got the T, the T, the T, the T, the T. But I didn't draw all those, okay? So here's one that got a T, here's that one got a T. So it's free to continue synthesis, correct? But a subset here stopped at that position next time. But this molecule in there had the T, so it was free to go. It got the G. It couldn't get the dideoxy G because it was not in that tube. Okay, this is one type of dideoxy per tube. That's different than the fluorescent, where there's 4 types, okay? Does that help? Okay. All right, so we get a trace like this. That's the trace you'll get. You'll figure out the sequence from it, okay? And again, remember, if you want to read this, it's CGAAGTCAG from the traces, and that's the strand being made. And then it's easy to figure out the template, okay? All right, so let's move on. Question. So the question, I think, is asking why does dideoxy terminate the reaction? Because there's no longer an OH to polymerase to. So we have to do it in this process because we have 4 separate tubes, and it's the termination that allows us to know this, okay? Without ever stopping, let's say we never saw this stopping pattern because we didn't have these 2 lanes. We stopped here, but we wouldn't know what was going on at the 1st, 2nd, or 3rd, or 4th position. So we have to have all 4 types in this protocol, okay? But when we use the fluorescent labeled, we get by with 1, okay? You'll have to just trust me on it, go back and look at it, okay? Last question, then we really need to move on to the Pleiades stuff. I can't hear you at all. So the question is, how would you calculate probability of termination, okay? If I had 50% deoxy and 50% dideoxy, what's the probability I get a dideoxy? 1/2, right? What if I had 1% and 99%? So it's The way to think of this is, let's say I have marbles in my pocket, okay? 10 marbles. 5 are red, 5 are white. What's the chance I'm going to pull a red marble? 5 out of 10, 1/2, right? If I have 301 marbles and 1 is red and all the rest are white, what's the chance I'm going to get the red? 1 out of 301. If I have 100 white, or let's say 99 white and 1 red, the chance is 1 out of 100. It's strictly the ratio of them. Is that okay? All right, let's move on. Now the tough stuff, the ploidy. The sequencing stuff is actually relatively straightforward when you sit down and draw it out for yourself, okay? I can't stress that enough. Anyway, ploidy, remember, we've talked about this in the very first lecture. Ploidy refers to sets of chromosomes. In most of our cells, they are diploid. 2 sets of chromosomes. Who'd you get 1 set from? Mom. Who's the other set from? Dad. You know, students always say mom first. I don't know why. Always mom. I do the same thing. Anyway, this is showing this for a human cell. This is, let's say, a skin cell or liver cell or what's called a somatic cell, a non-gametic cell. And there's 46 chromosomes. There are 23 types of chromosomes. There are 2 sets. Chromosome type number 1, chromosome type number 2, chromosome type number 3, 4, 5, up to 22. And the last type, which we call the sex chromosomes, an X and a Y. Those are types of chromosomes. Okay? And at this chromosome number 1 is very similar to chromosome number 1 from Dad. There are differences along positions. Those positions are called loci. So when we talk about genes, in this pair of chromosomes, at this position or locus, let's say that green band there, there's still only 1 gene, but the information may or may not be different between the 2 chromosomes. Those actual pieces of information are called alleles. So when we say there are 23,000 genes in the human genome, that means there are 23,000 genes in 1 set of chromosomes. If we have 2 sets, we still have only 23,000 genes. We've not gained any genes, but we do have 2 alleles for each gene. Do you see the subtle difference? Genes referring to the locus. So for example, you talked about desulfonate 3-phosphate dehydrogenase. That's encoded at a position. I don't know where, so I'm going to make it up. Let's say it's at this green band there. So you had 1 allele from mom and 1 from dad, but you still only have 1 gene for that enzyme. But they could be different alleles, okay? And that's oftentimes confusing, trying to do my best to make it clear. So we have info from mom, we have info from dad. Along this chromosome, there are stretches that gives rise to information. Most people define a gene as a stretch of DNA that will eventually encode for mRNA, which will eventually be translated to give you a protein, okay? That's how most people define a gene. It encodes for mRNA that will be translated to form protein. I don't like that definition. I prefer the definition unit of function, because there are many pieces of DNA that are extremely critical that never encode for mRNA, never get translated into protein. But they're extremely critical, and we talk about those genetic differences between people. So if we talk about genetic differences, then they must be genes, okay? So anyway, piece of information. And you'll learn more about that. Promoters, piece of information, doesn't give rise to a protein product. Operators, doesn't give rise to a protein product. Anyway, a mutation means something's happened to the protein that is no longer functional, which is reflected by a change in the RNA, which is reflected by a change in the DNA. Since the DNA is the permanent molecule in all of this, when we talk about mutations, we refer to the DNA, and of course that information is then conveyed to the RNA, which is then conveyed to the protein. But genes are DNA, mutations are changes in the genetic structure of the DNA, okay? And of course, resulted in mRNA and then the protein. Any questions about that? All right. So here's, we'll be working with yeast. Yeast are amazing things. They can go from haploid to diploid states, and they can undergo mitosis in either state. That's not true for humans. Gametes cannot undergo mitosis. Gametes must fuse to form a zygote to undergo mitosis. That's why they're called gametes. When you can have a haploid cell that can undergo mitosis, it's called a spore. So like when you learned in botany and Bio 1B about spores, they can undergo mitosis to become 8-celled, megagametophyte, those sorts of things. Remember that? It's gametophyte because it produces a gamete. All right? So with yeast, we can either have haploid strains or diploid strains. Yeast that are haploid are great to do genetics on because we don't have to worry about a second copy of information. There's only 1 set. So it's really easy to do this. We'll work with haploid strains to make diploids, to get them to mate. They touch. I mean, I don't know what to describe their behavior, but they touch, they mate, okay? Simple as that. Now, they have to be of opposite mating types. A with alpha. If you have A with A, it doesn't work. Alpha with alpha doesn't work. But A and alpha, they mate. Okay? So what we're going to be doing is looking at haploid mutants. So we know they're defective in the biosynthesis of histidine, which is an amino acid. So if you can't make histidine, can that organism grow? No, because proteins have histidine in them. Some proteins do, okay? Almost all do. So since we have defects in there, if we have a diploid, we have 2 sets, but if we have haploid, 1 set of information. So this is just showing you a pathway. I've made it as easy as possible. A prime is a chemical. It gets converted to B prime, to C prime, to D prime, E prime. These are chemicals, substrates, or products, however you want to do them. And the conversion is catalyzed by an enzyme. And we're going to say this enzyme is encoded by 1 gene that codes the mRNA, then encodes the protein, okay? So 1 gene here, 1 gene there, 1 gene there, corresponding information. But remember, the DNA gets transcribed and then translated, and the protein's out there in the cytosol, okay? Doing whatever it's doing, all right? So let's look at this. If we have a pathway like this, and somehow there's haploid only, so one set here. Here's the starting molecule, here's the end. I would never give you the molecule of histidine on the exam and actually identify it. I would never give you this molecule and actually identify it, or draw the intermediates in here. All I need to know is this is an amino acid and this pathway, okay? And down here, in this conversion process, if I have a genetic defect that I can't convert D prime to E prime, what would build up in that cell? What chemical would build up? There's a block between DNA. What would build up? D. Just like if I had 1,000 students down here at the front of the room, and these are steps. I'm taking 1 step in each pathway, right? Converting. If I have a block here, all those students end up here. They can't get past that block, right? That's the block. So just like with the pathway, D would accumulate. Now, if somehow I could get these students moved from this block past that somehow,